We've conserved significant lands throughout these watersheds and trying to do what we can to maintain watershed health that way. We've really been expanding our work with partners to actually try to improve watershed health, not just keep it from getting worse. And Lisa Cleckner has been one of the folks who's been helping us find our way, because we are certainly not experts in this and we're learning like everyone else. And I've had the privilege of working with Lisa these past few years. Lisa is the director of the Finger Lakes Institute at Hobart and William Smith College. She joined Hobart and William Smith in 2010. Before that, she was the assistant director of operations at the Syracuse Center of Excellence in Environmental and Energy Systems. She has both a PhD and a master's of public health in environmental and health sciences from the University of Michigan. She also has an MBA from the Simon School at the University of Rochester and a BA in biology from Colgate. And her strength is this combination of collaborative aquatic research and community outreach, which really, I think, positions her well to kind of address this issue we're talking about today. So really appreciate, Lisa, you being with us tonight. And thanks so much for taking the time and look forward to hearing. Oh, Lisa.
Thanks, Andy, very much. Yep. And thanks, Edie, so much for helping with all the logistics. Really appreciate this opportunity to talk to some people that we don't get to talk to every day, and then also see many familiar friends and faces on this. So I think it's going to be a good night. And so I'm going to share my screen and go from there. And I'll look for a little affirmation that we are looking okay before I get too far down the road. So here's this screen and full screen. Does that look good?
Looks good.
Thank you. I think I know that voice. Okay. Well, good. So for tonight's talk, I'm going to go back and do a little bit of a review. It's been a few years since we've had a chance to be together. And I know some of my friends on certain lakes will have seen some of these slides, but I think a lot of it speaks to how we as a Finger Lakes region has really tried to look at harmful algal blooms, kind of how, when they started, but then also some of the scientific research and community programs that have evolved since we've started to learn more about it. And then looking at the future is, where, obviously we'll kind of end up. So here's a brief outline of where we are, and I will do my best to provide enough details without going into too much scientific detail, but I hopefully will be able to answer questions at the end if something is not clear, just because I have the chat on a different screen. I don't think I'll be able to get to it probably till the end, so bear with me, but please feel free to unmute and interrupt me if something's not working. So here is our outline for the evening. Okay, so I think Andy set this up really well. I mean, the lakes, we know that the lakes are really the heart of the economy of our region. Our two biggest economic drivers are tourism and agriculture, and water is, just so related and supports everything about both of those. And the big thing that I always like to mention is the ecosystem services that these lakes provide. It's a lot of the, not only things that we can put a monetary value on, but there's many things that the lakes provide to us that we can't really monetize, or certainly monetize easily. So thinking about culture and just being in a beautiful location and having the resources that we have, such as great fishing and amazing drinking water and wonderful habitat, both on land and in the lakes. And I think everyone that is on this call knows that the Finger Lakes really is a global brand at this point. And you can't pick up a publication a lot of times without finding something about the Finger Lakes in it. So the bulk of our presentation then is going to be talking about this threat to the Finger Lakes. And, as Andy said, HABs, harmful algal blooms, or cyanobacteria. There's a lot of different names for them, but I think if we take the word HABs and kind of break it down, the key aspects of this phenomena is basically that you have something that is harmful in the water. I'm an environmental health scientist. One of the things that I've always been interested in are threats to water quality. And we know that toxic compounds are a threat, and key to understanding the extent of the threat is understanding what the dose of something is, as well as what the concentration of it is. So taking those things into consideration, but then also considering what are some sensitive populations, or if it's your body, what parts of our body do we need to worry about being affected. And cyanobacteria affect a number of our systems, including the liver and skin, as well as nervous systems. The algae, I'll just go back to my Colgate days. Back when I was at Colgate, they were called algae more so we're just going to go with algae, but really they are cyanobacteria. And then the blooms really shows that it's in high concentrations. And so this is really kind of, I'd say, be exemplified really in 2014, a national crisis for the city of Toledo that just hit the national news. And this picture up on the right here shows Lake Erie, and this is a water intake crib that's in Lake Erie that brings the water into the city of Toledo for treatment. And this is what the raw water looked like in the lake. And this picture just always astounds me to be able to say, oh my goodness, that's a lot of cyanobacteria in there. And you can also see these blooms from space. I mean, remote sensing is a big part for understanding the extent and fate of these blooms. And typically what we're most concerned about are our drinking water supplies because of the presence of the toxins. But there are treatment technologies available for drinking water systems for a lot of the municipalities. But we also have to think about what happens when we're recreating in the nearshore waters, if our pets, such as dogs or children, are splashing water, being able, basically getting exposed to these toxins, trying to have a better understanding. And the conventional wisdom that talks about when these HABs happen usually call for about 4 ingredients. So they're typically in the sun, and that's usually because that's when most people are out recreating, to be honest with you. But typically there's little wind, so the water's calm, so these blooms can form on the surface. There's warm temperatures, and there's enough nutrients to enable the cyanobacteria to grow. So if we look at the New York State definition of a HAB, and this has evolved over time, so I put a couple of different figures on here. So on the left is really like the criteria. So the way that we look at them, cyanobacteria are one of a type of phytoplankton, which I'll talk about in a little bit more. But the key is what the concentration of that cyanobacteria community is. And there is a device called a fluoroprobe that can assess the blue-green algae chlorophyll in basically 3 minutes and tell you the concentration. And if you look at the New York State criteria, basically you want to be, in order for it to be considered bloom, so high enough cyanobacteria concentration, the blue-green algae chlorophyll concentration has to be greater than 25 micrograms per liter. Then if the samples exceed that, then they are tested for toxins on top of it using looking for microcystin. And then there's a couple of criteria, one, whether it's open water or shoreline. And so then you can get basically a bloom that's then confirmed with high toxins, and that would be a HAB. So that's a fairly sophisticated structure that the state used to identify and call a HAB. And with all of the participation and all the people, I'd say, looking for HABs, they learn that people, when they see a HAB, they know what a HAB looks like. And you don't necessarily need this device, but it's very helpful to confirm the concentrations, as we'll see later on for some of the research that's going on. But the state, a couple of years ago, went to basically an online reporting system, NIHABS, and you can send a picture in, somebody will look at it and it looks like a bloom, and they're going to say, yep, it's a bloom. And the advice is to know it, avoid it, and report it. And so the monitoring system has kind of evolved over time. So if we look at our Finger Lakes, this is just an overview of data from the last 11 years. And I've got the 11 Finger Lakes listed going from west to east. And you're going to see a box here going, from left to right for each year that observations were made about the presence of cyanobacteria on the lakes. Now, if something is essentially a light blue box, it means that there was a bloom, but there's no information about toxins per se. If there's a red box, then those were confirmed high toxin events. And so you can see, if you kind of look at all of the lakes, really in the early 2010s, right, there were a few lakes, Owasco and Honeoid, that were seeing some of the HABS early on. But as we progressed and maybe more people started learning about them and just kind of keeping an eye out for them in the fall or the summer when they typically appear, then more lakes started seeing them. And then these red boxes essentially confirmed that high toxin events were also measured. So I think this big kind of line in the sand, if you will, is probably this 2016 going into 2017, when a lot of the lakes that typically are considered to be low nutrient lakes, such as Canandaigua, Skaneateles, and Keuka, had harmful algal bloom events. And, HABs were always thought to be, well, it's only going to be the lakes like Honeoid or Owasco that tend to have a little bit higher nutrient concentrations that are going to have these events. But as you can see, it's a full, every single Finger Lake is seeing them now. And with the advent of that new program where the state stopped toxin analysis, then you can see the number of events that were reported for each lake in the table, but there were no toxin values measured for those. So that just gives a snapshot as to, it's definitely been trending, unfortunately for us, in the more HABs and more lakes as time moves on. So taking half a step back, here's really a timeline of the whole New York State HABS program going back to before 2011. And, there's been a lot of attention, I'd say, at the state level to this question. And, the 2014 event here in Toledo, but then the DEC is working with parks. But I think a couple of the big events that got a lot of the public's attention, certainly in the Finger Lakes, was this toxin detection in Owasco Lake of finished drinking water, which happened in the fall of 2016. And in 2018, there was also one on Canandaigua Lake from a public drinking water supply system. So these are, certainly things that municipal drinking water systems need to pay attention to. And then we also need to think about the health of people that might not be part of a municipal system and are taking drinking water directly from the lake. And what do we do with that? So, this is a concern for all of us. And then, there's obviously the recreational aspects of this as well. So I always start this with, and sorry for those of you who have seen this probably in the last couple of weeks, but this cyanobacteria, it's an enormously large topic area, and I'm going to try to just give a little bit of information about how well these organisms have done over time and how they've survived, and why they're a problem. In 2002, there was a book published about them. It was 669 pages, and there have been just addenda, chapters that have been added to it since then. And in 2012, another basically volume of this, 760 pages, and again, with more chapters added to it. So, these cyanobacteria are organisms that have been around for basically as long, 3.5 billion years old. And so I'm a biologist, I have a really hard time with this time scale. But to think of Earth, and cyanobacteria were the dominant life form for over 1.5 billion years. So they are responsible for us having basically oxygen to breathe. If we go back to high school biology, high school chemistry, these bacteria are able to photosynthesize. They have pigments in them that allow them to basically, to take carbon dioxide and with light, and they can make sugar and oxygen. And with that metabolism, they created oxygen for all of us to breathe in the geologic time scale. They're found in virtually every habitat on Earth, from the hot springs to the polar regions, symbiotic relationship with lots of plants. And, as a phytoplankton or primary producer, they're the base of the food web. And so really taking the sun's energy and, basically providing food for upper trophic levels. So cyanobacteria are just one type of phytoplankton or algae that are part of the natural community of phytoplankton. And so if anybody's ever been lucky enough to have a microscope out on a lake, you get to see these incredible organisms that live in the water all the time. And you probably have heard of things like diatoms. They have silica structures, they're frustules, they have chlorophyll in them, so they're photosynthesizing as well. But diatoms tend to have a peak in the spring and in the fall. And once the water warms up, green algae show up. So these are some desmids and some like colonial green algae. There's some little smaller phytoplankton, cryptomonads, some dinoflagellates, some golden algae, denobrion. There's just these incredible organisms, like I said, once you get them under a microscope. But the blue-greens are also a part of that natural phytoplankton community. And so they're always there. It's just that they tend to peak when the temperatures get warmer. And that's typically for us, obviously in the August and September time period, water temperatures. So with their 3.5 billion years of experience of living on Earth, they've really developed a number of, I call them survival strategies, but it's really just they're being who they are, right? And so this is I have a house on Cayuga Lake, and I'm standing on my dock in September and looking out. It looks like a beautiful blue day, kind of looking forward to hanging out on my swim platform. And then I started looking a little bit more closely to the water, and what you can see here are these cyanobacteria starting to get where the water's starting to get cloudy here. And then as the sun goes forward, you can see, there's some nice eelgrass there, great fish habitat, I should say. But the cloud, you're just like, okay, something's not right. And one of the things that the cyanobacteria can do is they can move up and down in the water column. They have gas vacuoles, and they position themselves optimally to take advantage of the best light levels and where the nutrients are. And with that, then they can grow prolifically, and then they can shade out other plankton as well. So not only are they finding the best spot, but then they can grow faster and then keep the other types of phytoplankton at bay. And a lot of times, we see them on the nearshore area, and then you get a gentle wind or just water that's moving towards the shoreline. You'll start to see these blooms concentrating on shore. And I think, in the Finger Lakes, this is a pretty common occurrence these days. So if we look at another way that the cyanobacteria they're basically built to succeed, is to look at the chlorophyll and the pigments that they use to basically convert the light energy into sugar or starch. And so their pigments, they have chlorophyll A in them, and this is basically meant to be the light spectrum going from blue light to red light. So if you think of like Roy Biv and just all the different wavelengths of light going from blue to red, from short wavelengths to long, chlorophyll A has a peak for absorption in the around 400 nanometers, and one way over near the infrared, or near red at 700 nanometers. They also have accessory pigments that can either harvest light under low light conditions, or sometimes they can provide photoprotection. And cyanobacteria tend to have phycoerythrin as part of their makeup, as well as phycocyanin. And you can compare this to like a green algae, where they would have a chlorophyll A, and which has the two peaks, and a chlorophyll B, and they typically have carotenoids as well. But it's a pretty narrow band of light energy that they can take advantage of compared to the cyanobacteria. The cyanobacteria really can capture energy across a much wider wavelength. So, just another good advantage for them compared to the other phytoplankton. The next thing that I want to talk about is their morphology, or a lot of times these are very large organisms. They're colonies essentially. So this is microcystis up here, and you can see like these single dots are individual cells, but they tend to form these really large organisms through colonies. And these are really hard to eat. If you think of the typical grazers or filter feeders such as zooplankton that are typically feeding on phytoplankton, these are enormously large to eat. And then because they have a mucilaginous sheath around them, they're typically not digested. So they can pass right through a digestive system and continue to grow. And you think of dry-scented mussels, zebra mussel, quagga mussels. A lot of times these are the cyanobacteria, they're spit out or just pass through without being digested. So another, I'd say, good advantage for the cyanos. And then we have the access to nutrients. They have the ability, certain genera of cyanobacteria have the ability to pull nitrogen from the air and actually use it as a nutrient. So when we think of nutrients that help things grow, NPK, right, is that, I think, for all the gardeners out there. So nitrogen is one element we need. Phosphorus is usually the limiting nutrient in lakes. But if there is enough phosphorus, then things can continue to grow, up to a certain level, then nitrogen becomes limiting. And so certain cyanobacteria have these specialized cells, they're called heterocytes, that they can actually take atmospheric and turn it into a nutrient that they can use for growth. And, so that can be converted into nitrate, urea, not into urea, sorry, but into ammonium. And so, if we look at nitrogen pathways, they're pretty complex transformations. But the important thing is that cyanobacteria can also pull nitrogen from the air. And so one of the things that we're going to talk a little bit about later on is just where these nutrients are coming from. And there's a lot of controversy, I'd say, in the scientific literature. There's been whether P is the limiting nutrient, and for most freshwater systems it is, but N also plays a role, and particularly with cyanobacteria, because of the role that the nitrogen plays in toxin development. And so if we want to take another look at then toxins, these are some molecular structures of toxin. And if you look at basically the backbone of a lot of these molecules, there's nitrogen that's involved with it. And there's been research that has shown that, while the phosphorus controls the amount of microcystis that you can have in a cyst, in a particular area, the amount of nitrogen is what controls the toxicity levels a lot. And there's a lot that we can talk about with, basically with toxins. And what makes it complicated with cyanobacteria is there's a number of different species that can produce toxins. Sometimes they have the gene for toxin production, sometimes they don't, and it can't be the same species. And then even if they have the gene, they don't always express the gene. So then you're getting into genetics, into molecular methods to try to understand better about why and how these organisms are producing toxins. And so that's a whole wonderful place for research. And so I want to talk a little bit more about some of the different work that we've done with watershed associations. And we have been doing this with a number of different partners since 2016 and picking up new partners all along. And this is showing the flora probe here in action. Essentially, we get a lake sample in from a partner, and it's poured off into a cuvette. It's put into this device, the fluoroprobe. We put a cover on it, and you have basically hit start, and it'll analyze the chlorophyll concentrations of 4 algal groups, namely the green algae, the diatoms, the cyanobacteria, and the cryptophytes. And so this is what some of you've seen pictures of these already, but these are some Finger Lakes cyanobacteria. And then we can measure toxins through a different method. And so I just want to bring, this is something that we did back in 2019 with Seneca Lake and Canandaigua Lake. Our community partners, basically lakeshore residents, and anybody working in municipal governments or anybody recreating, invited to go out and find blooms, after receiving training from the DEC. And, looking at, this is Canandaigua Lake. This was in 2019. They had 28 zones that they monitored, 26 volunteers, 36 miles of shoreline. Seneca Lake, 85 zones, 100, you can read the stats on this. These lakes are really, really big. I mean, it's one of the things that, you sometimes don't appreciate how big they are, but if you can imagine trying to sample at every single location, this would be impossible for scientists to do. So basically people keeping an eye on their own lakeshore areas, and when they see a bloom, bringing it in, so that's what we worked with the partners to do. And we did these cyanobacteria chlorophyll measurements. So I'm going to show a little bit more data here from 2019, because I think it's important to show that I talked originally about the 25 micrograms per liter of the blue-green algae chlorophyll that the state considers to be a bloom. And you can see this is Canandaigua Lake here on the left, and this 25 micrograms per liter is a solid line going across here. And you can see pretty much every bloom, or almost every sample, exceeded that concentration. And the scale goes up to 1,400 micrograms per liter. On Seneca Lake, same thing. Their blooms tended to start a little bit later, but you can see the concentrations of chlorophyll exceeded 100,000 micrograms per liter at times. So very, very high concentrations of these things. And the other thing that I want you to pay attention to is some of these bars have a little bit of red on them. And so we were trying to understand when these red bars show up as part of the chlorophyll signal, because most of the other chlorophyll is all cyanobacteria, what's going on when those red ones are there. And so that was part of a kind of a mini research project with our partners to better understand that. And just wanted to put some summary tables on here. Essentially, the state, because, I'll just say there was a cap on the number of blooms that the state wanted to run for toxins, just because this is expensive to do. And, it's also, if you think of every single lake in New York State, I think there's over 15,000 water bodies in New York State. It's a tremendous amount of work. And there was a couple of key people in Albany that were running this whole program. But the important part here is that there were a lot of blooms on these lakes, some very high chlorophyll levels. These were, when a bloom was suspected, there were blooms. And of the, basically 55 samples that we had, and then 25 of those were analyzed for toxins, a number of them were toxic. And so trying to better understand, are we going to have a high toxin event or not? We were trying to get a better handle on it going back to 2019. So I'm going to skip this one just in the interest of time, but we could tell what that little red bar was on the chlorophyll peak, and it had to do with different species or different genera of the cyanobacteria that we were seeing in the water at the time. I'm happy to go back to the site later if needed. But one of the things that we learned as we were going through this is when you see a HAB, and this is, unfortunately, this is my doc here from September of 22. So it was a very thick bloom. And you can see that there's, in this image over here, there's a lot of I'd say heterogeneity in the bloom. And you could really introduce a sampling bias by trying to collect a very thick sample. And this is and I tried to take a very, very thick sample. But this is from a scientist's perspective, it's hard to say, okay, well, what is, a HABS sample? Like, what's the exact technique in order to have a better understand? Because we wanted to quantitatively get a better handle on what these events look like. And so that transitioned into a project that we ended up doing with Canandaigua Lake Watershed Association and the council, as well as Cornell. We received funding through the Water Resources Institute, and our main goal in this project was to say, okay, can we go out and, we know what a bloom looks like, and we know when we see a bloom, it's usually probably going to be a high toxin event just because of, the data that has been collected over the years. So the key with this particular study was like, can we just go out and collect a water sample off a dock and, basically have it be a 0.5 meter composite sample. So, sticking a PVC pole into the water and collecting, you get about 300 mils of sample. So what is that about? So 0.5 liter is 16 ounces. So, pretty close, about 10 ounces maybe. So you get about 10 ounces per collection in this pole sampler. And we collected samples 0.5 meter from the below the surface and 0.5 meter off the bottom of the lake. And we did this in near shore areas. So this is some volunteers that we worked with on Canandaigua Lake, and they started pole sampling with us starting in July, and did it every week on a Tuesday, because we wanted to just say, okay, what is the natural variation of water, chlorophyll, and toxin concentrations as we go forward? So we assessed that with the fluoroprobe. We did some microscopy. Cornell has a project that's, we did some toxin analysis using the ELISA method, which is the EPA standard method for that, a certified method. And then Cornell was also doing some assessment of the cyanobacteria to see if the genes that could produce toxins were present in the samples that we were collecting. And then also looking at the community analysis of the other bacteria that are part of the water sample, because cyanobacteria just don't, they're not the only things present in the water column. We wanted to have a better idea with that. And we also work with the Community Science Institute. They did some parallel work on Cayuga Lake. And so Ruth is a faculty member in the Cornell Engineering School. So with this, I'm just going to show you all the data for the season. We worked with Canandaigua for about 14 weeks, 12 weeks, and Cayuga had a slightly different schedule. But the key thing here is I want you to look at this x-axis. I'm sorry, the y-axis of these are 4 location surface and bottom samples, and these are the chlorophyll concentrations that we saw off of Canandaigua Lake using those pole samples. So remember, a bloom is 25 micrograms per liter. We saw concentrations over 1,000 micrograms per liter for blooms in 2019. But every single pole sample, the range of the y-axis here goes up to 6 micrograms per liter of chlorophyll. So very low chlorophyll concentrations. Cayuga has more nutrients in it, definitely showed some higher chlorophyll concentrations through the season. So we, surface and bottom as a general rule, were not that different from each other, but we did this week, every single week. Canandaigua had HABS samples that year as well. Probably, well, here's 1,000, here's 2,000 micrograms per liter. So kind of a typical HABS season, moving forward, this starting in, kind of middle August going into September and October. And we measured toxins, and the high chlorophyll samples had high toxins. So then what we wanted to do was to say, okay, can we measure the toxin concentrations even in just straight up water, and how do they change over a season? And so the way we did that is we looked at the microcystin concentrations relative to the amount of chlorophyll that we are capturing, so that you basically get a toxin quotient per amount of chlorophyll. And, does that toxin concentration change over a season? And what's pretty remarkable about this, and here's a ratio of 1, if you were on this line, you'd say, well, you'd get 1 microgram per liter of microcystin relative to 1 microgram per liter of chlorophyll. And we saw a very similar relationship in the pole samples with very, very low concentrations of chlorophyll and toxins, but it showed the same seasonal pattern, and definitely peaked, kind of in the September timeframe, which they both peaked around the same time. The reason that some of these weeks don't have HABS samples is because there weren't HABS samples that week, but we were able to look at that ratio in the pole samples. So if you compare this to Canandaigua, if you compare Canandaigua to then Cayuga and to Seneca Lake, as you go through a season, so one would be starting like in late June and going all the way till, basically late October, Canandaigua Lake definitely showed a higher microcystin concentration relative to chlorophyll. And so we found this to be really quite fascinating. And so we wanted to, we're still exploring why this is the case. And I know Canandaigua Lake Association has some additional projects going on to continue to explore this. But the big part of what we were trying to do with this project was to look at the rest of the microbial community that is part of bloom. And if you think of like a microbiome, if you think of your gut basically, and you think of all the healthy bacteria that are there, it's the same thing with water samples. There's bacteria that's just part of everything. So we're trying to understand what's the relationship between the cyanobacteria and the other groups of bacteria. And this is all done through molecular methods at Cornell. And we're still exploring this, but as has been reported in other systems, there's 3 most prevalent phyla of bacteria that happen to be around as part of the water samples. And so this is an area of continuing work. So I just wanted to show a little, hopefully this is going to work. This is a water sample, and this is taken with our microscope. And this just shows you what we see when we put these things under the microscope. So this floating thing here is a colony of Dolichospermum. And these are Vorticella. And these are organisms that are attached to it, but they can move as well. So these colonies of organisms not only have other, organisms that are associated with them, but then like this cyanobacteria up here, Microcystis, also has other cyanobacteria lodged into its mucilage. So these communities are very complex. So when you take a water sample, it's not just, there's nothing, you might see a particle or 2 in the water, but you put them under the microscope, there's a lot happening. And so we're trying to get a better handle on this particular aspect of the microbiome. So looking ahead and just trying to sum up a little bit, there's been a lot of work, and, I'm just highlighting some of the small work that we've done, but we know a lot of other partners have done a lot of work, like Untitled Atlas, Syracuse has done some, it's an ESF, there's so many other institutions, and we're trying to work together when it makes sense. But some of the work that we've been doing recently on Owasco Lake, and Owasco Lake here, we did some sediment incubations. So we've collected sediment cores to try to better understand, like, where if these nutrients aren't, it doesn't rain every single day. So are they coming in from the watershed, or are they actually coming out of the sediment? So we set up these incubations with cores. We have filtered surface water that we run over the cores, and we measure nutrients that are given off by the cores. And this is oxygenated water, so it's not meant to be like the deepest part of the lake. But are there nutrients being released all the time from the sediments? We just want to have a better handle on this. And so I'm not going to go too much into the numbers, but it depends on the season, and then it depends on the site. But we found, and I picked on soluble reactive phosphorus here, which is the most bioavailable form of phosphorus for phytoplankton. And we found that for site F, which was up here at the northwest corner of the lake, that there was a lot of SRP coming out of the sediments in June of 2022. Now, if you look at these same 4 sites later in August, and I apologize because these are different y-axes, so it's a terrible science graphs, but I wanted to just show the numbers because you never would have seen these otherwise. The flux of phosphorus was actually negative, so it was actually being taken up by the sediments in August, except for at this very southern site down here, which had 3 milligrams per square meter per day taken up or being released, and that was compared to 29 in June. So we're trying to just get a better handle on this. And like I said, there's a lot of people trying to study the role that sediments are playing. So if you think of large storm events and things like that, where there's water column mixing or upwelling, it's like what's happening even in these nearshore areas, not even in the deepest part of the lake, that could be contributing to nearshore nutrients. So then that brings us a little bit to climate change and kind of just looking ahead. We know that the temperature of our lakes are increasing, at least in the Finger Lakes. We know the CO2 concentrations are up, and our precipitation patterns are changing. This is a picture that one of my neighbors on Cayuga Lake took in February of 2017. There's a big farm field behind us, and there was no snowpack, and there was a big rain event. And you can see that the fields are inundated. If nothing's growing there, nothing's taking up that water. And then all of that, clay / phosphorus laden water is just coming right into the lake through storm, pipes that are basically trying to keep people's houses from flooding. So we need to figure out how to manage this water. The other thing is for cyanobacteria, as the temperatures get warmer, they like it hotter than the other cyanobacteria or than the other phytoplankton types do. So as our temperatures continue to increase, they're going to continue to do better and better. And just wanted to end a little bit with this whole aspect of the low nutrient lakes and HABs. And this is very much an emerging area of research. I mean, again, even I think when I was in graduate school, everyone was like, yeah, it's high nutrient lakes only. But low nutrient lakes are also seeing HABs, including lakes like Lake Superior. And so here's a paper that was released. It's a couple of years old now, but it's an international author list. So this is a global phenomenon. We're seeing it more and more. And some of the species that are genera that we see of cyanobacteria across the globes, it's like, well, what do the cyanobacteria do well? They can regulate their buoyancy. They can, essentially control how fast they move up and down the water column. The thick filaments prevent organisms from eating them, or if it happens to actually dry out, they can actually prevent desiccation. So, a lot of the things that we've already talked about, these cyanobacteria groups are already conditioned to survive and do quite well. So some other things that came up in this paper is that the cyanos compared to the other phytoplankton groups like the greens and the diatoms, they can store excess phosphorus and nitrogen better. And so if those nutrients become limiting, the cyanos they've already got some kind of sitting in the refrigerator, if you will. They walk over and open the door and continue to eat, but it's all within their own physiology. They're also capable of taking up organic forms. If we talk about carbon very little, but there's also some competitive advantages that cyanos have with respect to carbon dioxide, the use of carbon dioxide and bicarbonate as part of its metabolism. And then just like what's happening with climate change and with these mixing and upwelling events, what about the hydrodynamics of the lakes, storm events, and just making sure that we're considering that this is not just high nutrient lakes, but it's really all lakes. And the cyanos are going to do quite well as we move forward in all lakes based on the changes that we're seeing in our environment. So with that, I just want to very quickly acknowledge many of our funding sources. This they all play a role in helping us have a better understanding of the Finger Lakes environment, and want to make sure that we're acknowledging them. I want to thank my colleagues at the Finger Lakes Institute. We have programs in education. We have a certified lab. We do a lot with invasive species. We host the Finger Lakes Prism. And we have 2 watershed managers on our staff, one for Seneca Lake and one for Cayuga Lake. And so we have a number of professionals that are out there doing work all the time. And I hope you see them working across our region, and all the things that we do are interrelated. We also work with a number of faculty at our own institution, as well as across a lot of the regional institutions. I'd like to thank them. And then, of course, our students, we learn a lot from them every year. They bring a new perspective. And one of the things that we hope that we can share with them is the beauty of the Finger Lakes region, and how lucky they are to go to school here, and how lucky we all are to live here and to be able to take, to enjoy the many assets of our waters and watersheds. So thank you with that. And I will leave you a little happier picture, I think, from my lake house, as opposed to the big harmful algal bloom that we showed from September. So thank you for your time, and look forward to your questions.
Great. Lisa, if I could start with one, I thought that was a great overview. And you mentioned climate change and nutrient runoff, but could you also share what is either known or suspected about the role that mussels, particularly quagga mussels, might play in the fact that I think many people have had my experience, where a lot of experience where never seen a bloom, and then 2017, bam, they're everywhere. And so I'm curious, just even if it's speculative, what your thoughts are on that.
Sure. I think, the quagga mussels they've cleared out a lot of the phytoplankton, right? They've made the lakes clearer. And so different organisms are able to grow on the bottom now than, like, if you probably have heard of Cladophora as well. So the algae that's growing on the bottom of the lake, particularly in the Great Lakes, they also have brought nutrients that used to kind of cycle out in the deeper part of the lake, through phytoplankton, zooplankton happening out there. They've brought it to the near shore area. So the cyanobacteria, are able to take advantage of nutrients that are cycling very closely to shorelines. And there's enough light that they can establish themselves as well. So, I mean, they definitely, I think if you look at like New York State as a whole, if you look at every lake, I know there's been some big studies that have been done that have shown that lakes that have north-south kind of alignments and have long fetches and have dry-scented mussels are the ones across New York State that tend to have the blooms. But really, the dry-scented mussels have fundamentally changed how nutrients cycle in all lakes where they're present, including the Great Lakes, and really changed food webs as well.
Okay, ready for some more questions?
Absolutely.
Okay, first one, why did New York State stop measuring the toxin levels? I think that was going back to the beginning of the presentation.
Sure. I think, and I'm just speculating, but I think, I mean, one of the great is that they figured out, like, once they saw a bloom, that it was high toxins. So, like, if we know that it's a really thick, high chlorophyll concentration, it's going to have high toxins if it's a cyanobacteria, according to the methods that are, the EPA certified methods. So why spend the money to do the analysis when you can pretty much say, yep, that's a high toxin event, and you really shouldn't be in the water anyway.
Can you discuss whole lake treatment options or mitigation techniques that may help the lake safer for recreation?
There's a lot of different mitigation techniques that have been tried. I will say the DEC has funded some of those things, and there's a good reference point to go and look at the mitigation page that the DEC has to see what has worked and what hasn't worked. Honeoye Lake is trying to, at this point, is doing an alum application, trying to keep the phosphorus that is already in the lake, in the in the sediments, and not have it be released anymore. So I'm pleased to see that New York State is trying some other methods, but they're hard to do. And I know like Owasco Lake has used ultrasonic nebulizers to try to put bubbles in the water to have the gas vacuoles disrupted so that the cyanobacteria can not regulate their particular location in the water column. So, I mean, I'm all for pilot projects to try to better understand them, but I think it's really hard to change whole lake systems once these things are here.
Okay. If nitrogen in the air during fixation can be somewhat unlimited, would phosphorus still be the main limiting nutrient in that regard?
It's a complicated question. It's not all cyanobacteria can fix nitrogen. That's probably one thing I didn't get into the particulars of. So like microcystis does not fix its own nitrogen. But I will say, microcystis has a lot of friends, other bacteria that do fix nitrogen. So these microbiomes, really work as a community and an ecosystem to try to do these things. But if you think of, as, basically organisms that could use phosphorus up to a certain point, then if you have the ability and all the phosphorus is gone, that then you could get nitrogen from a different source. From the air, then that there is a competitive advantage there. And there's a lot of chemistry that goes along with nitrogen just because it is cycled through the air. There's a lot of transformations similar to what happens to phosphorus in the sediment, where you can get reduced forms of nitrogen that are more bioavailable, as we like to call it, so that they can incorporate it more easily into their metabolism. So yes, there's a lot of ongoing work on that particular topic. But typically, if you ask the state, phosphorus is limiting nutrient. That limits the growth, but some other things like the toxin production are definitely related to nitrogen as well.
Are there any methods in use using hyper/multispectral remote sensing to map the affected areas?
That's a huge field. There's been a lot of work on, basically the coastal areas of the United States for, like, if we think of brown tide and red tide and things like that. But there's also a big push to better understand inland lakes and what's happening with chlorophyll detection. And I know, I think ESF is doing some of this work. We're part of a project with SUNY Fredonia, Binghamton, and Corning that is using hyperspectral cameras mounted to drones to try to better understand what's happening. There's a lot of, it's complicated when you're going from space, and doing monitoring down to the water level, and especially when shorelines are involved, HABS, and it has to do with pixel size and some of the interferences you get when you're taking pictures of something that has land in it and water and a bloom in it. So, but we've made a tremendous amount of progress, I would say, we being the larger scientific community, as to having better understanding of how we might better use those going forward. Because the citizen scientists are fantastic, but not everyone's sitting out on their dock or their house, like looking out at the lake all day, right? And blooms they're very ephemeral, they can come and go. And so anything that can capture images on a large spatial scale and, obviously time is very useful.
Okay, did I understand correctly that Canandaigua had a less diverse phytoplankton community? Why would that be? And is there any research that suggests a lack of diversity increases toxin production?
I'm not sure I said that there was a less diverse phytoplankton community. It might have been in relation to the other bacteria that are present, and it could just be due to the absolute numbers because there's fewer, there's lower chlorophyll levels in Canandaigua, and presumably when you're measuring bacteria too, there's just going to be fewer organisms that are present in order to understand what the community is. So I'm not sure we can really make any inferences about the diversity of it. It was just, what we found in some of the beginnings of our molecular methods to better understand the types of bacteria that are present in the lake.
Concerning wind and water agitation, are windy and wavy conditions acting on cyanobacteria by dispersing colonies and thereby lowering colony counts and toxin levels by some action of oxygenation by depriving colonies of optimal sunlight? Let me know.
I think the wind and the wave energy definitely helps to mix them in. And so, I mean, it kind of puts everyone on equal footing again. There's more nutrients that are available. I think that's definitely one of the best ways to disperse them, right? Is to wait basically for it to become a little bit windy and they will blow offshore. So, but that's, I'd say you need to still be aware when they were just like there like an hour ago, and just, make sure that you're taking appropriate cautions if you do decide to go into the water. But definitely those help to distribute them throughout the rest of the water column and to other parts of the lake.
Okay, last question. Last summer, I did not see a bloom by my house on Canandaigua Lake, but there were a lot of dots in the water. How dangerous are they?
So I think if you look at the World Health Organization, they have some pretty strong recommendations on this. And, everyone always has to assess their own susceptibility and sensitivity of this. But if you, I think they use the example, if you can stand in the water and you can see your feet, it's not terrible. But, so those few dots, I would pay attention to them. You don't want them to get worse, but it's not like it's going to, be the worst thing that you've probably ever seen for exposure. I was just going to say, I mean, I think that the DEC criteria, right? If you see it, report it. A lot of times there's things like pollen that can look like HABS, but I mean, people that have been doing this for a number of years know when it's pollen season. They know what's, flowering at that point. So we've learned a lot from the different things that can also kind of be the cyanobacteria lookalikes, if you will. So it's great.
Lisa, there's one last question that popped up. Someone is interested in collaborating with interested parties to provide detection and solutions via drone technology. Do you have any specific contacts that you can suggest for that?
If somebody wants if they can send me an email, and I'd be happy to connect with the Corning project. And I can also, share with other lake associations and, other university collaborators that we might know of. But it's definitely an emerging area and people want to know more about that.
Okay, great. That's all the questions. So thank you very much, Lisa.
Great. Thanks so much, Lisa. And thanks everyone for joining us tonight.
This evening.
Have a good evening, everyone. Thanks for joining us.